Okay, so let's start the next lecture. Is the CR present? Is the CR present in the class? Okay, so if you are the CR, please send me an email. Okay, thank you. Um, so Mr. Jasem, if I'm not pronouncing it incorrectly, please send me an email. Please send me an email after the class and introduce yourself, and I will provide you with the PDFs of my slides. Okay? Okay. Okay, so in this lecture, I'm going to talk about microbial growth, cultivation, metabolism, and genetics. Basically, we will be talking only about bacteria. So here are the objectives of this lecture. First of all, we will be talking about bacterial growth. What do we call the bacterial growth? We will be talking about cultivation, minimum requirements for bacterial growth. Also, we will be talking about molecular oxygen requirements for bacterial growth. Also, we will discuss what do we call a growth curve, about the metabolism, about the mechanisms that bacteria use to generate ATP. Also, we will be talking about the structure of bacterial genome and gene exchange in bacteria. So let's start with bacterial growth. Bacterial populations grow through an increase in the cell number. So increase in the size or volume or length of the cell is not called a bacterial growth. Okay, so when we talk about bacterial growth, we consider bacteria as a population. So when the number of bacteria in this population increases, we say that this population is growing. So we don't see bacteria as individual cells when it comes to bacterial growth. So prokaryotic cells, including bacteria, they reproduce through cell division, and binary fission is the most common type of cell division in bacteria. During the binary fission, first of all, a cell, which we call them a parent cell or mother cell, elongates, its chromosome is replicated, and it is obvious because the cell is going to become two cells, and each cell needs the chromosome, so it makes sense that the parent cell first of all start to replicate its chromosome. So the cell elongates a little bit, a chromosome is replicated, and two chromosomes migrate to opposite poles or opposite ends of the cell, and then a septum, which is a cross wall, starts to form in the mid cell. And when the formation of this septum is completed, it splits the cell into two identical daughter cells. We call them identical daughter cells because they have identical genetic material. But sometimes the DNA polymerase, which is responsible for replicating the DNA, it makes mistakes. And if this happens, we see that the genetic material of these daughter cells are a little bit different. But without any mutation, without any DNA polymerase mistakes, we see that we have two identical daughter cells. The time which is needed for a bacterium to double its numbers is called a generation time, or doubling time, which is related to the genetic information of this bacteria. Some bacteria, they have just a very short generation time. They are able to double their numbers in a few minutes. But some bacteria, they have a very long generation time. Some bacteria, they need some days, several days, to be able to double their numbers. So it's related to the genetic of bacteria. We're able to cultivate or propagate bacteria in the laboratory. So why do we need to cultivate bacteria? Do you have any idea? Why do we need to cultivate or propagate bacteria in the laboratory for diagnosis, for tests like urine culture, the type? You know, when a patient suffers from an infectious disease, physicians collect a specimen from the patient, from the infectious site, and send it to the microbiology laboratory to identify the causative agent. So I told you that it's important for physicians to know the exact agent which has caused this infectious disease, to manage the patient, to actually start a therapy. And the medication that they are going to prescribe for their patients is related to the type of the infectious agent. So it's really important to identify the infectious agent. And for this purpose, first of all, you need to take the specimen from the infectious site, and the site that you think that the infectious agent is present there. And you take the specimen from that site, you send it to the microbiology laboratory there. Uh, the workers in the laboratory, they are able to cultivate the infectious agent. They are able to multiply the number of the infectious agent to the level that now they are able to run the tests. Okay, so for running the test to identify the microorganisms, for bacteria, you need to have a sufficient amount of them to run the test. So that's why we need to cultivate bacteria in the laboratory. And sometimes we just cultivate bacteria in the laboratory because we are interested in them. We want to study them. Okay, so there are different reasons that we need to cultivate or propagate bacteria in the laboratory. So, based on the physical properties of the culture media, there are three main types of culture media. We have solid or agar culture medium, we have liquid or broth culture medium, and also we have semi-solid culture medium. We can divide culture media based on different criteria, but based on the physical nature, we have three types of culture media. So agar, it's a type of a polysaccharide that we add to the culture medium, to the liquid culture medium, and it's able to solidify the culture media. And we have the solid culture media in this special container that we call Petri dish, as you can see in this picture. And you're seeing some spots on this type of culture medium in the left image that you see in this slide. And the spots that you are seeing, they are colonies. I already told you about colonies. Each colony, each spot that you see on this plate is the result of growing of only one bacterial cell. And as you can see, these colonies, they have different colors. So it means that they are different from each other. And as you can see, they have different shapes and they have different sizes. And as I told you, the macroscopic appearances of a bacteria can be used to classify them. And here you're seeing an example. And in liquid culture medium or broth culture medium, there is no agar. And in semi-solid culture medium, there is agar, but the concentration of agar is less than solid culture medium. So basically, we have three types of culture media when it comes to the nature of culture media. So as shown in this slide, bacteria have minimum requirements for growth. So if you want to grow bacteria in the laboratory, you need to provide them with whatever they need to grow, okay? So here we're going to learn about the minimum requirements that you need to provide with bacteria to grow. When bacteria enter into the body, they know, I'm sorry. So when bacteria enter into the body, they know how to use the body, our body, as nutrient. But when we grow bacteria in the laboratory, we need to provide them with some requirements. Well, of course, all bacteria need an energy source. Okay, they are living organisms, so it means that they need an energy source. Medically important bacteria, the bacteria which cause disease in human, they use organic molecules like, for example, sugars as the energy source. So different bacteria have different energy sources, but medically important bacteria, they use organic molecules as the energy source. Example is, sugars. Another minimum requirement for bacterial growth is a carbon source. Also, they need a nitrogen source and various ions. When we examine a bacterial cell, we see that it is largely composed of carbon and nitrogen, and that is why these sources are essential for bacterial growth. And as you can see, we have classified them as the minimum requirements for bacterial growth. Also, certain ions are critical for bacterial growth. For example, potassium, sodium, calcium, magnesium, chloride. These are important ions that bacteria need to have in order to grow. Carbon, oxygen, hydrogen, nitrogen, phosphorus, and sulfur, they are essential elements. Especially we talked about carbon and also nitrogen sources. And also there are some components which are needed for the activity of the bacterial enzymes. We have some elements here. We have zinc, we have cobalt, we have manganese, we have, for example, copper, and also we have iron here. Iron is particularly very important, because, when bacteria enter into the host, when they enter into the human body, you should know that the free iron concentration in the body is very, very limited. So when they enter into the host, they must acquire iron from these low concentrations. The bacteria, they produce these specialized proteins which are called siderophores, as you can see in this slide. And siderophores are able to scavenge iron. They are able to bind to iron even in very low concentrations, which we have in the body. And once they are bound to iron, they get back to the cytoplasm of bacterial cell and they release the iron. And this is how some bacteria are able to get iron when they enter into the body, by using the siderophores. So siderophores are small proteins. Bacteria secrete them into the host, and they are able to bind to iron even in the low concentrations, and when they are bound to iron, they get back to the cell, and this is how bacteria are able to get the siderophore. Also, remember that during the inflammation, when the bacteria are present in the body and when they cause inflammation, the body also further reduces the free iron as a defense mechanism. Okay, so this is one of the defense mechanisms that we have in the body. So when there is this inflammation in the body tries to lower um, the, the, the amount of free iron that we have in the body. And the purpose is to limit the access of bacteria to the iron. But when we are trying to grow bacteria in the laboratory, we need to make sure that we have added iron in their culture medium, okay, because we want them to grow. So the siderophore that I told you, it is true in the context of when bacteria have entered into the host, when they want to cause disease in the human. And bacteria are living microorganisms, so it makes sense that we say that they need water also to grow. And regarding molecular oxygen, it is not like that all bacteria they need molecular oxygen. We humans, yes, we need molecular oxygen to grow. We need molecular oxygen to stay alive. And this is not true about all types of bacteria. Some bacteria, yes, they need molecular oxygen for their metabolism to grow. But molecular oxygen is toxic for some bacteria. So if they are exposed to molecular oxygen, they will be dead. So as you can see, if you want to grow bacteria in the laboratory, you need to make sure that if they need molecular oxygen, you need to provide them with the oxygen. If they don't need molecular oxygen, you need to remove the molecular oxygen from the environment so that those types of bacteria are able to grow. So based on the oxygen or molecular oxygen requirements, bacteria are categorized into 5 groups. First of all, we have obligate aerobes. They need molecular oxygen for their metabolism and growth. An example is Mycobacterium tuberculosis. Mycobacterium tuberculosis. It's the causative agent of tuberculosis. So we talked about Mycobacterium species. What do you remember about Mycobacterium? What do you recall? We mentioned Mycobacterium in the previous lecture. Um, Mycobacterium species, they do have a cell wall. The one that you are referring to is Mycoplasma. This one is Mycobacterium. Staining was different, exactly. Um, they have different Mycobacterium species. They have different, cell wall. As I told you, they have this waxy cell wall, which makes it very hard for stains to penetrate into the cell wall. So that's why we can use gram staining technique for them. We can use them. You cannot use gram staining technique on them. If you want to stain them, you need to use acid-fast staining technique. So we have this bacterium, Mycobacterium tuberculosis. Okay, so this one is responsible for tuberculosis. Have you ever heard the name tuberculosis? Yes. Can you tell me more about tuberculosis? What is tuberculosis? It's a respiratory disease. It's contagious. Yes, it's a contagious bacterial disease. The patient who's suffering from tuberculosis, they cough. Sometimes it's a bloody coughs. They have fever. They lose weight. Sometimes the Mycobacterium tuberculosis is highly resistant to antibiotics, and it's possible that you lose your patient. So this bacterium, anyway, it lives in the lung, and so Mycobacterium tuberculosis is an obligate aerobe, and it's the causative agent of tuberculosis. So obligate aerobes, they need molecular oxygen for their metabolism and growth. As I told you, an example is Mycobacterium tuberculosis. So let's move on. Second, we have obligate anaerobes. As you can judge by their names, they cannot tolerate molecular oxygen. Molecular oxygen is toxic for them. They don't need molecular oxygen for their metabolism. And if you want to grow them in the laboratory, you need to make sure that you have removed all the molecular oxygen from their environment. Otherwise, they will be dead. They die. For example, we have Clostridium perfringens here, which is a gram-positive bacterium. It causes gas gangrene, which is a type of soft tissue infection. So this bacterium is a Clostridium. So I told you that Bacillus and also Clostridium genera, they are able to form spores. So it means that this bacterium, Clostridium perfringens, is also able to form spores. Sometimes the spores of this bacterium are present in the environment. If someone has a wound and the spores of this bacterium just get access to the wound, they start to germinate, and they cause gas gangrene, which is a soft tissue infection. We will be talking more about gas gangrene. So the third group of bacteria when it comes to molecular oxygen requirements are facultative anaerobes. These bacteria can grow with or without molecular oxygen. For example, we have Escherichia coli here, which is a gram-negative bacterium. We talked about, gram-negative bacteria. I told you that Escherichia coli is a very famous bacterium, and this bacterium is a facultative anaerobe. It's able to grow in the presence and also absence of molecular oxygen. And next we have aerotolerant anaerobes, and these aerotolerant anaerobes, they are anaerobic bacteria, so it means that they don't need molecular oxygen in their metabolism, but they are able to survive in the presence of molecular oxygen. So in that case, they are different from obligate anaerobes. Obligate anaerobes, they are very sensitive to molecular oxygen. If they are exposed to molecular oxygen, they die. But aerotolerant anaerobes, these are also anaerobic bacteria, but they can tolerate molecular oxygen. They don't die if they are exposed to molecular oxygen. Example is Clostridium tertium, which is a gram-positive bacterium, and it can cause wound infection. Wound infection. And finally, we have aerophils. These groups of bacteria, they need molecular oxygen, but only at low concentrations. An example is Helicobacter pylori, which is a gram-negative bacteria. Facultative anaerobes, they are able to grow in the presence and absence of molecular oxygen, okay? So they don't care if the molecular oxygen is present or not. So I told you that we have different types of culture media that we use in order to grow bacteria in the microbiology laboratory. And I told you that we can classify culture media based on their physical nature. And also, there are four types of culture media that you can see in this slide. We have enriched non-selective media, we have selective media, we have differential media, and also we have specialized media. Specialized media, they are used in order to grow a specific or a special type of bacteria. So when you will study bacteria in your future, sessions, you will be learning about some specialized media which are designed only to support the growth of a specific group of bacteria. So first, let's talk about the first type of culture media, which are called enriched non-selective media. Enriched non-selective media, as you can judge by their name, they contain nutrients which are able to support the growth of most bacteria. So they are non-selective, okay? There is no inhibitors inside this type of culture medium which inhibits the growth of bacteria. There are, enough nutrients which is able to, support the growth of most bacteria. That's why we call them enriched non-selective media. A good example is blood agar. You're seeing a plate which contains a petri dish or a plate which contains a blood agar here. There is this basal culture medium inside this, container, which is supplemented with blood. So basically, we add sheep blood into this type of culture medium, and we have blood agar. So blood agar is able to support the growth of many bacteria because there is no inhibitors inside this type of culture medium. And we have another enriched medium, which is called chocolate agar. Let me type it for you. So we have chocolate agar. So chocolate agar also is another enriched non-selective medium. It also contains sheep blood, but we have heated this type of culture medium, okay? So that's why we call this chocolate agar, because the color of this type of culture medium is darker. It's like brown. It has this brownish color. So it contains blood, but it is exposed to heat. Okay, so we have heated. If you heat the blood agar, you have the chocolate agar, and again, it's another enriched non-selective media that is able to support the growth of many bacteria. So another type of culture media are selective media. Selective media, they contain inhibitors that suppress the growth of unwanted microorganisms. So there are some inhibitors inside this type of culture medium which inhibits the growth of a specific group of bacteria. For example, we have McConkey agar. You're seeing the McConkey agar here. It's a selective culture medium. There are some inhibitors inside this type of culture medium which inhibits the growth of, gram-positive bacteria. So it allows gram-negative bacteria to grow on this type of culture medium. So McConkey agar is a type of selective culture medium, it allows only gram-negative bacteria to grow. It inhibits the growth of gram-positive bacteria. In contrast, we have another culture medium, which is called mannitol salt agar. This is also another selective culture medium. It is selective for Staphylococcus species. You know, Staphylococcus, it's a gram-positive bacterium. You will be learning more about this bacterium in your future lectures. Just know that this gram-positive bacterium is able to grow on this type of culture medium. But this medium actually inhibits the growth of other bacteria because this type of culture medium contains a high salt concentration, as you can judge by its name. And this high concentration of salt inhibits the growth of most bacteria, but the Staphylococcus is able to tolerate this high concentration of salt, and that's why it's able to grow in this type of culture medium. So selective media, they contain an agent which inhibits the growth of unwanted bacteria. And I told you about the McConkey agar, and also I told you about the mannitol salt agar. Also, we have differential culture media. Differential culture media are, supplemented with inhibitors to actually suppress the unwanted growth of microorganisms. The McConkey agar that we have again here, it is a selective culture medium. I told you that this is a selective culture medium because it contains, some agents which are able to actually inhibit the growth of gram-positive bacteria. So only gram-negative bacteria are able to grow on this type of culture medium or McConkey agar. So that's why it's a selective culture medium. It's also differential culture medium. It means that there are some ingredients inside this type of culture medium which actually differentiate between bacteria. McConkey agar contains lactose, which is a type of sugar. Some bacteria are able to use lactose in their metabolism, some bacteria don't use it in their metabolism. If the bacterium is able to use lactose in their metabolism, they produce acid in their metabolism, and the acid actually decreases, it drops the pH of the culture medium. And if this happens, the color of the colonies changes to pink. For example, we have Escherichia coli here. Escherichia coli we talked about this bacterium, is a gram-negative bacterium, so that's why it is able to grow on McConkey agar. So McConkey agar is a selective culture medium. It inhibits the growth of gram-positive bacteria, so only gram-negative bacteria are able to grow, like Escherichia coli. Also, it is a differential culture medium, so it means that there are some ingredients inside this type of culture medium, and these ingredients help to differentiate between bacteria. For example, Escherichia coli here is able to use lactose in its metabolism. So it means that it's able to decrease the pH of the culture medium, and if this happens, it changes the color of its colonies to pink. So if you see pink colonies on the McConkey agar, it means that your bacterium in question is a lactose positive. So it means that your bacterium has been able to use lactose in their metabolism. And if you don't see pink colonies on the culture medium, it means that your bacterium is not able to use lactose in their metabolism. And you can just differentiate bacteria based on the ability to use lactose in their metabolism or not. So we talked about this, that we can just classify bacteria based on the fact that if they are able to use a specific type of sugar or other nutrients or not. And this is a very good example to that case. So here you can just distinguish between bacteria based on the fact that if they are able to use lactose in their metabolism or not. So differential media, they contain ingredients, and by using those type of ingredients, you can differentiate between different types of bacteria. A good example, again, is Meconic agar, which is a selective and also, you know, differential medium. So here you can see the growth curve of bacteria. This growth curve actually shows the number of viable cells in a bacterial population over time. So if you add bacteria to a new culture medium, and if you wait some time, and if you measure the number of living cells inside this type of culture medium, and if you plot the data that you get, you will be having such graph, which is called the growth curve. And as you can see, there are 4 phases in this curve that you have get. The first phase is called lag phase. So as you can see in lag phase, there is no increase in the number of living cells. So during the lag phase, bacteria are just adjusting to the new environment. They are just adapting to the new environment. So we don't see an immediate cell division when we add bacteria to a culture medium. First, they need to adjust to the new environment. And then they enter into the log or exponential phase. During the log or exponential phase, the cells are just dividing rapidly. And after a while, they get to the stationary phase. So the third phase of the growth curve is called the stationary phase. And as you can see, there is no increase in the number of living cells here. The reason for this is that limitation of nutrients, exhaustion of nutrients. Bacteria have been growing inside this culture medium. They have been using the nutrients in this type of culture medium. So now they have exhausted the nutrients inside the culture medium. And that's why we don't see the increase in the number of living cells. And then another fact in this phase is that the waste products have been also accumulated inside the culture medium. So when bacteria are growing, they also producing some byproducts because of their growth. And these byproducts are toxic for bacteria. So during the stationary phase, the concentration of these waste products has increased inside the culture medium, and that's why these waste products have negative effects on bacterial growth, and that's why we don't see increase in the number of cells. And finally, we have death phase or the decline phase. And during this phase, most cells stop dividing, and bacteria are also dying during this phase. Some bacteria just don't die; they just stop growing, and they become very resistant to antibiotics. So during the death phase, some bacteria are dying, as you can see, and some bacteria just don't die. They just stay viable, but they don't grow. And in that case, they are very resistant to medications. So let's get back to the log or exponential phase. During this phase, bacteria are, very susceptible to specific types of antibiotics, including penicillin. We talked about penicillin. Penicillin is a type of antibiotic which I told you that interferes with the cell wall synthesis. And when bacteria are growing, they are, synthesizing the cell components, including the cell wall. And it means that bacteria are most susceptible to penicillin because they are growing, they are synthesizing the cell wall, and penicillin is also resistant. So penicillin interferes with the cell wall synthesis, and that's why bacteria are most sensitive or susceptible to penicillin during their life or exponential phase. And let's get back to the stationary phase. If the bacterium is able to form spores, they do this during the stationary phase. I told you that the limitation of nutrients actually triggers the spore formation. And here in the stationary phase, we have limitation of nutrients, and that's why bacteria start to form spores during this phase. Can you define metabolism for me? Thank you. So metabolism is the total of all biochemical reactions which happen within a cell, and it is divided into two main parts: catabolism and also anabolism. So during catabolism, large and complex molecules are broken down into smaller and simpler molecules, and this part of metabolism is called catabolism, and energy is released because of, molecules are breaking down, and the energy which has stored inside the bonds are getting released, and this energy is used in anabolism. So during anabolism, larger and more complicated molecules are synthesized from smaller and simpler molecules. And even the macromolecule cell structures are built during the anabolism, and this process actually needs energy. So we have synthesis in anabolism, and synthesis needs energy. For example, here we have an energy source which is glucose. If you take a look at the left part of this cell, it is showing you the catabolism. So we have this energy source. Most bacteria are able to use glucose as the energy source. So the glucose is broken down during the catabolism, and as you can see, the energy is getting released, and the energy is used in anabolism. The energy is used to synthesize, the subunits, the macromolecules, the cell structures. All of these processes, they need energy, and the energy is coming from catabolism. Medically important bacteria, the bacteria which are able to cause disease in human, they are able to generate ATP through three mechanisms: aerobic respiration, anaerobic respiration, and also fermentation. In aerobic respiration, bacteria use molecular oxygen as the final electron acceptor, and they generate a large amount of ATP. In anaerobic respiration, bacteria use other molecules other than molecular oxygen as the final electron acceptor, and they produce less ATP in comparison to aerobic respiration. And finally, we have fermentation. And in fermentation, there is no electron transport chain. I told you when I was telling you about the electron transport chain that there are some bacteria, they don't have the electron transport chain at all. So here in this slide, you can compare aerobic respiration, anaerobic respiration, and also fermentation. So let's start with aerobic respiration. As I told you, most bacteria are able to use glucose as their energy source, and that's why I have used glucose to teach you these mechanisms. So microbiology books use the glucose when they want to teach you these mechanisms. That's why I'm also using glucose to teach you these mechanisms. So first of all, in aerobic respiration, consider that there is one molecule of glucose as the energy source. So first of all, glucose undergoes glycolysis. The most common type of glycolysis used by bacteria is Embden-Meyerhof pathway. So during glycolysis, some of the energy which was stored in the chemical bonds of the glucose is released. So as a result, two ATP molecules are produced. Also, two NADH molecules are also generated. NADH is a molecule which has the potential to produce ATP when it donates its electrons to electron transport chain. So for now, when one molecule of glucose enters into the Embden-Meyerhof pathway, some of the energy which was stored in the glucose is getting released. So we have two ATP molecules and two NADH molecules are also released. So at the end of glycolysis, we have two pyruvate molecules. Each pyruvate molecule has 3 carbon atoms. So glucose has had 6 carbon atoms. Now it is converted into 2 pyruvate molecules. Part of its energy is released. And then these 2 pyruvate molecules need to actually enter into the Krebs cycle. So Krebs cycle is needed to extract the remaining energy from the glucose. But before entering into the Krebs cycle, each pyruvate molecule undergoes a preparatory step. Each pyruvate molecule loses 1 carbon atom as CO2, transfers its electrons to form NADH. So as you can see, 1 NADH is released during this preparatory step. And also it binds to coenzyme A. So as a result, 2 molecules of acetyl coenzyme A are formed from 1 glucose molecule. So each acetyl coenzyme A enters the Krebs cycle. As we had 2 pyruvate molecules, so it means that we have 2 acetyl coenzyme A, and each acetyl coenzyme A enters 1 Krebs cycle. So during each turn of the cycle, 1 ATP, 3 NADH, and also 1 FADH2 molecules are produced. In Krebs cycle, 1 GTP actually is released, but GTP equals ATP when it comes to the energy. Okay, so that's why we say that in each cycle of Krebs cycle, 1 ATP molecule is released. Also, 3 NADH and a 1 FADH2. FADH2, like NADH, is able to donate its electrons to the electron transport chain, so it also has the potential to produce ATP. And also two CO2 molecules are released. Each acetyl coenzyme A has two carbon atoms, and these two carbon atoms get released during the Krebs cycle. So the NADH and also FADH2 molecules, which are released during this pathways and also during the Krebs cycle, they need to donate their electrons to the electron transport chain. In aerobic respiration, the actually final electron acceptor is molecular oxygen. And in aerobic respiration, we get the greatest amount of ATP from one molecule of glucose compared to, anaerobic respiration and also fermentation. The reason is the molecular oxygen is the best electron acceptor compared to other, molecules. That's why we get from one molecule of glucose, we get 38 ATP molecules in aerobic respiration. So in anaerobic respiration, glucose also undergoes glycolysis followed by the Krebs cycle, which are similar to aerobic respiration. The key difference here is the final electron acceptor in the electron transport chain. So instead of molecular oxygen, in anaerobic respiration, other molecules act as the final electron acceptor. Sometimes there are some bacteria which use nitrate, some bacteria use sulfate, some bacteria use carbonate as the final electron acceptor. And all of these molecules that I told you, they aren't as good as molecular oxygen when it comes to accepting electrons, and that's why we get less amount of ATP molecules compared to aerobic respiration. So I told you that in aerobic respiration we get 38 molecules of ATP, but in anaerobic respiration we get less amount of molecular ATP. We get less amount of ATP. So this is the electron transport chain. I told you there is no mitochondria in bacteria. Electron transport chain, if it is present, it resides in the cytoplasmic membrane of bacteria, and this is the electron transport chain. It's a series of complexes in the cytoplasmic membrane of bacterial cells. NADH and FADH2, they donate their electrons to this chain, and the electrons flow down, and their energy is used to pump protons or H plus out of the cytoplasm of the cell to the outside the cytoplasmic membrane, as you can see. So the energy of the electrons is used to pump out the H plus or protons. And finally, the electrons are received by molecular oxygen. Molecular oxygen has this high electronegativity, and it serves as the final electron acceptor in aerobic respiration. So it is an ideal molecule for efficient ATP production. In anaerobic respiration, as I told you, there are other electron acceptors instead of molecular oxygen, and as I told you, they aren't as good as molecular oxygen when it comes to accepting the electrons. They have lower affinity for electrons, and that's why we get fewer ATP molecules from each molecule of glucose during the anaerobic respiration. From each NADH molecule, we get 3 ATP molecules, and from one FADH2 molecules, we get only 2 ATP molecules. And as I told you, the energy of the electrons is used in order to pump out, the H plus or proton from the cytoplasm to the outside the cytoplasmic membrane. So it means that now we have this high concentration of H plus outside the cytoplasmic membrane, and we have this electrochemical gradient now. When these H plus ions enter into the cytoplasm through ATP synthase, as you can see in this picture, their energy is used to generate ATP. So basically, the energy of electrons is used to generate this electrochemical gradient, and this high concentration of protons, when they enter into the cytoplasm, they release energy, and their energy is used to generate ATP. And this is how basically ATP is synthesized. And the last mechanism that I'm going to tell you about, which is used to generate ATP inside the cell, is fermentation. So some bacteria get their energy, they produce ATP by using fermentation. Again, we have one molecule of glucose which undergoes glycolysis. And during this process, 2 ATP molecules are produced, also 2 NADH molecules, and 2 pyruvate molecules. And that's it. The only number of ATP molecules that bacteria get from fermentation is 2. So bacteria only get 2 ATP molecules from fermentation of 1 molecule of glucose, and as you can see, they get very low amount of ATP from 1 molecule of glucose. There is no Krebs cycle, there is no electron transport chain in fermentation. Okay, so bacteria, some bacteria just don't have the electron transport chain in their cytoplasmic membrane. So as there is no electron transport chain in fermentation, NADH cannot donate its electrons there. So it means that NAD plus is not going to be regenerated. All the NAD plus sources of the cell will be used by the glycolysis, and all the NAD plus of the cell will be converted into NADH. And it means that finally the glycolysis will stop working because there is no NAD plus to enter into the glycolysis pathway. So it means that the glycolysis will stop working. There is no ATP getting generated because the pathway is stopped, and the cell will be dead because it's not able to get ATP from glucose anymore. There is no NAD plus in the cell to enter into the glycolysis pathway. So to solve this problem, instead of transferring or donating the electrons to electron transport chain, NADH molecules, they donate their electrons to organic molecules inside the cytoplasm of the cell, only to regenerate NAD+. Remember that during the fermentation, when NADH molecules, they donate their electrons to organic molecules, this process is not used to, get ATP like at all. Okay, they just do this in order to regenerate NAD plus. I'm sorry, they do this only to regenerate NAD plus. So, depending on the type of the electron acceptor inside the cytoplasm, which is an organic molecule, we have different types of fermentation. For example, we have lactic acid fermentation, we have ethanol fermentation, we have other types of fermentation in bacteria. So, based on the organic molecule, which is receiving the electrons from NADH molecules, we have different types of fermentation processes. So we have lactic acid fermentation, we have ethanol fermentation, we can have other types of, you know, fermentations. Okay, we talked about the electron transport chain also. Now, let's talk about bacterial genetics. So bacteria are diploid or haploids. Bacteria are haploids. Good. So it means that they have only one copy of chromosome inside their cells. So bacteria are haploids. So I'm going to tell you more about the structure of bacterial genomes. Now, bacteria, they have a single, and most of the time, a circular molecule of DNA as their chromosome. Some bacteria, they have extra chromosomal genetic elements, which include plasmids and also bacteriophages. In the left image, you can see the plasmids. Plasmids, in comparison to chromosomes, they are, smaller DNA molecules. And, most of the time, plasmids are also, circular DNA molecules, but there are exceptions. There are some bacteria which have, linear chromosome instead of circular chromosome, and also linear plasmids. I don't want to go into the details, just know that most bacteria, they have circular chromosome. If plasmids are present, they are also most of the time circular. Sometimes, the plasmids, they contain genes which encode proteins, and these proteins are able to give resistance to antibiotics, give resistance to antibiotics. So by having those plasmids, bacteria show resistance to specific type of antibiotics. And also the thing is, plasmids can transfer from one bacterium to another, so it means that that bacteria can spread the antibiotic resistance between them. So that's why the presence of plasmids are very important in the clinical context. And also, sometimes there are some genes on plasmids which are involved in the, disease causing process of bacteria. So this is how, this is why plasmids are very important. About bacteriophages, bacteriophages are viruses which infect only bacterial cells. So bacteriophages, or simply just phages, they have nothing to do with the human cells. They only infect, they only target the bacterial cells. Here, in the right image, you can see the structure, a typical structure of bacteriophages. Bacteriophages, they have different shapes, okay, but this one is a typical one. So they have this protein shell, or they have this membrane, and inside this protein shell, there is the genome of the bacteria, which could be DNA or RNA, and The bacteriophage actually attaches to a bacterial cell and it injects its genome into the bacterial cell. And after injecting its genome, sometimes the bacteriophage genome start to replicate inside the bacterial cell. And the structural proteins of the bacteriophage are also synthesized inside the cell. And genome and the structural proteins, they find each other, so the genome gets packaged inside the bacteriophage, and complete bacteriophage particles actually start to build up inside the bacterial cell. And finally, they kill the cell and gets released into the environment, and they can find another host, and this cycle actually repeats. So this type of bacteriophage infection is called lytic infection. It is called lytic because the bacteriophages have lysed the bacterial cell. They have killed the bacterial cell. So that's why this type of infection is called the lytic infection. But sometimes bacteriophages, they cause the lysogenic state. So it means that bacteriophages, they don't start to replicate immediately when they inject their genome into the bacterial cell. First of all, their genome just integrates into the bacterial genome, and it can stay there even for a long time. And when there is this stress, when there is this damage to the bacterial genome, the bacteriophage genome actually exit from the bacterial genome and start the lytic infection. So it means that it start to replicate its genome, and it start to synthesize its proteins. The genome gets packaged inside the protein structure of the bacteriophage. The cell will be filled with new bacteriophage particles, and the cell will be dead. The cell will be lysed, and the new bacteriophages get released into the environment, and again they can find another host to repeat the cycle. So some some bacteriophages, when they inject their genome inside the bacterial cell, they cause the lytic infection. Their genome start to replicate immediately. But sometimes bacteriophages, they cause lysogenic states. What is the definition of mutation? Can you define mutation for me? Any change in the sequence of DNA is called mutation. So any change in the sequence of DNA is called mutation. There are several types of mutations. Some of these mutations are shown in this slide and also the next slide. So as you can see in this picture, there is this DNA sequence. There is no mutation in this DNA sequence. It is transcribed into mRNA, and it is translated into a protein. So now let's talk about the situation that there is a mutation in the DNA. One mutation is called transition mutation. Transition mutation happens when one purine is replaced by another purine, or one pyrimidine is replaced by another pyrimidine. Purines and pyrimidines, as you already know, are types of nucleotide bases. A and G are purines, while T and C are pyrimidines. In contrast, in transversion mutation, a purine is replaced by a pyrimidine, or a pyrimidine is replaced by a purine, as you can see in this picture. Can you define missense mutation for me? I don't know if you are familiar with different types of mutations, but can you define missense mutation? Mutation which leads to change in amino acid reading, single nucleotide base in DNA is swapped for another one. You know, in missense mutation, as you can see in this picture, there was a serine amino acid in the protein, and now it's replaced by a leucine. Okay, so as you can see, there is a change of amino acid in the amino acid sequence of the protein, and it is called the missense, as uh, you told me correctly. So a missense mutation is a mutation in which the DNA sequence changes, and that results in a different amino acid being added to a protein. This happens when a codon is changed, and the new codon actually codes for another amino acid now. And you know the definition of codon. Codon is a sequence of 3 nucleotides that codes for an amino acid. There are two types of missense mutations: conservative missense mutation and also non-conservative missense mutation. In conservative missense mutation, the new amino acid has similar properties to the original one. Maybe the new amino acid, which is replaced with the original one, is having similar properties. For example, both amino acids are acidic amino acids. Okay, so it means that maybe there is no drastic change in the function of the protein because the new amino acid has similar properties with the original one. But also there are some non-conservative mutations. These types of non-conservative mutations, they are also missense mutations, but the new amino acid has different properties compared to the original one, which can affect the protein's structure and function. Maybe the new amino acid is a basic amino acid, but the original one was an an acidic amino acid. So as you can see, the new amino acid has different properties compared to the original one, and that's why it affects the function and the structure of the protein. So sometimes mutations are nonsense mutation, as you can see here. Because of a nonsense mutation, a codon which encodes an amino acid now is converted into a stop codon. So it means that the ribosome falls off the mRNA. And because of that, we have a premature termination of translation, and we have a truncated, which most of the time is a non-functional protein. So we have nonsense mutation. So in nonsense mutation, one codon which encodes an amino acid is converted into a stop codon. And we have silent mutation. In silent mutation, a nucleotide is replaced by another nucleotide, but as you can see, there is no change in the sequence of amino acid in the protein. Although we have a change in the sequence of nucleotides in the DNA, there is no change in the um, sequence of amino acids in the protein. This happens because of the degeneracy of the genetic code. As you know, some amino acids just are encoded by multiple codons. And it is possible that because of the mutation, one codon is replaced by another codon, and both of these codons encode for only one amino acid. And in that case, the change in the sequence of nucleotides in the DNA does not result in the change of amino acid sequence. Another mutation is frame shift mutation. A frame shift mutation results from the insertion or deletion of nucleotides in number which is not divisible by 3. This, this actually disrupts the triplet reading frame of the mRNA, which leads to the altered downstream amino acid sequence. You know, in most cases, we have a premature stop codon, which results in a truncated and also non-functional protein. So frame shift mutations, as you can see, changes the sequence of amino acids from where the mutation happens. So after the mutation, downstream the mutation, we have different amino acids compared to the original one. And as I said, most of the time, the ribosome faces a premature stop codon, and we have we have a short protein which is non-functional most of the time. And we have null mutations. They are very dangerous for the cell. And these types of mutations completely abolish the gene function. Null mutations happen when there is this extensive insertion, deletion, or large-scale chromosomal rearrangement. So that's why I told you that null mutations are very dangerous mutations because they, abolish the gene function. Mutations can happen spontaneously in nature. I told you that, sometimes DNA polymerase, which is responsible for, replicating the DNA, it's not error prone. Okay, sometimes it makes mistakes, and if this happens, we have mutation. There are some physical and also chemical agents which are mutagens, which means they are able to cause mutation in the DNA. In this slide, I'm going to talk to you about the physical mutagens, which are heat, ultraviolet light, and also ionizing radiation. Heat actually promotes the amination of nucleotides. Ultraviolet radiation actually induces pyrimidine dimer formation. Take a look at the image that you see in this slide. We have a double-stranded of DNA, and this DNA is exposed to ultraviolet light. Now, ultraviolet light has energy, okay? And this energy is absorbed by pyrimidine bases that we have in the DNA. And it damages the pyrimidine bases. And they become very reactive when they are damaged. And if there are two damaged pyrimidine bases adjacent to each other, next to each other, on one strand of DNA, they react with each other and a covalent bond forms between them. So this is actually called the pyrimidine dimer. And these pyrimidine dimer, they interfere with the normal function of DNA polymerase. So if there are too many, you know, actually pyrimidine dimers in the DNA, the cell will be dead. So we will be learning about the function of ultraviolet as an agent that we use in order to destroy microorganisms. And this is the reason behind this function of ultraviolet, because it's able to form pyrimidine dimers, and pyrimidine dimers, they interfere with the normal function of DNA polymerase and they cause mutation. And if there are too many mutations in the DNA, the cell finally will be dead. So this is the action of ultraviolet, which is a physical agent as a mutagen. And also we have ionizing radiation. We have ionizing radiation. Example is X-rays. Ionizing radiation, they are very powerful radiations. They have energy enough to, damage, molecules, and they produce actually reactive radicals, free radicals, which are very reactive. And these free radicals are able to, damage macromolecules inside the cell, including the DNA that we have in the cell. So X-ray actually is able to break down the DNA, the double-stranded of DNA. They can, break down only one strand or double two strands of the DNA. And we have chemical mutagens. Chemical mutagens are divided into three major classes. We have, nucleotide base analogs, we have frame shift mutagens, and also we have DNA reactive chemicals. So let's start with nucleotide-based analogs. As you can judge by their names, they resemble the normal nucleotides, and they can, incorporate into the DNA, which leads to the mispairing during the replication of the DNA. For example, we have 5-bromouracil, which is a which is an which is a nucleotide analog, and it's similar to T, nucleotide T that we have, but instead of pairing with A, it pairs with G. And that's why, because of this, chemical agent, a TA base pair is converted into a GC pair. So as you can see now, there is this mutation in the DNA. So frame shift mutagens are, polycyclic molecules. They are flat and they are polycyclic. Example is acridine derivatives. They actually intercalate between the base pair in the DNA helix, as you can see in this image. And actually it disrupts the normal spacing between the base pairs, and it can result in insertion or deletion of nucleotide during the replication. So basically this changes that we see in the spacing between base pairs makes the DNA polymerase to make mistakes. So as I said, they make DNA polymerase to insert or delete only one nucleotide during their replication, and that's why we have frame shift mutation because of these types of chemical agents. And the last group of chemical agents are DNA reactive agents. Um, they actually alter the chemical structure of DNA bases. For example, we have alkylating agents such as, the example is nitrosoguanidine. These alkylating agents are able to add alkyl groups to the bases, and because of this change in the chemical structure of the DNA bases, we have abnormal base pairing, and sometimes these changed nucleotides can't just pair like at all. So that's why they are able to add mutations in the DNA. And the last topic that I'm going to talk to you about in this lecture is about gene exchange in bacteria. Gene exchange in bacteria can happen through several mechanisms, including transformation, conjugation, and also transduction. Let's start with transformation. You can see the transformation process here. Transformation happens when a bacterium takes up naked free DNA fragments from its environment and integrates them into its own genome. So we have this donor cell, which releases its DNA after it's dead. And we have this recipient cell, which is also known as the competent cell, which takes up these fragments and incorporate them into its genome. It's possible that the new fragments of the DNA which this complement cell is receiving, it's possible that they contain a new gene that the recipient cell doesn't have. So it means that because of the transformation, now the recipient cell is showing new features because now it has new genes which are coming from the donor cell. And it's important to know that not all bacteria are naturally competent, so it's not the case about all bacteria. It's not like that all bacteria are able to uptake the naked free DNA from their environment. That's not true. Only some specific strains of bacteria can uptake the DNA in this way. And in those groups of bacteria, they become competent at the end of their log phase of growth curve. As you can see, you're seeing a growth curve here. And as you can see, at the end of the log phase or exponential phase of the growth curve, bacteria are able to uptake the free DNA from the environment. So another way for gene exchange in bacteria is called conjugation. We talked about sex PLS or FPLS. I told you that it's a special type of PLS which helps bacteria to exchange genes. So here we have a cell which contains a plasmid. This plasmid is having genes which are able to encode the PLS. Okay? So a PLS or a sex PLS forms on the cell which has a plasmid. This cell is called the donor cell. So the donor cell actually encodes a special type of PLS which is called sex PLS or FPLS. Basically, this sex PLS forms a physical bridge between donor cell and the recipient cell. During the process of transferring the plasmid through the sex PLS, a single strand of plasmid is transferred into the recipient cell, and the synthesis of the you know, complementary strand is happening inside the recipient cell. So only one strand of the plasmid DNA is transferred through the sexpulus from the donor to the recipient cell. And I told you that sometimes plasmids they may have genes which are responsible for antibiotic resistance, or sometimes there are genes which are responsible for disease causing. And as you can see, by conjugation, bacteria are just transferring those genes to other types of bacteria. They are just spreading these genes. And that's why I told you that plasmids are very important when it comes to clinics. And the last method that bacteria use in order to, transfer their genetic element is mediated by bacteriophages. They are viruses. I told you that infect only bacteria. And sometimes bacteriophages, they capture fragments of bacterial DNA during their infection, and they package them into their viral particles by mistake. And when they infect the new host cell, they just transfer those fragments of DNA from their previous host to the new host. And this DNA can integrate into the recipient's genome, and it's possible that this fragment encodes a new gene, which the recipient cell doesn't have. And in that case, the recipient cell is showing new features. Transposons are jumping genes. They are mobile genetic elements. They can move DNA within a cell. They can actually jump from one genomic location to another genomic location or between DNA molecules. For example, they can jump from one plasmid to a chromosome, from a chromosome to a plasmid, and this process happens inside the cell. So the simplest transposons are called insertion sequences. And insertion sequences are very short. They contain, as you can see in this image, they contain inverted repeat sequences at their ends. They carry only the essential genetic information which is required for transposition. They encode the enzyme which is called transposase. Transposase is the enzyme which is responsible for, transferring the transposon between different DNA molecules or inside uh, one DNA molecule from one location to another. Sometimes we have some complex transposons. They contain additional genes, and it's possible that they have, they may contain, antibiotic resistance genes. And in that case, they are able to transfer those genes, for example, from a chromosome into a plasmid. And I told you that plasmids can transfer from one bacterium to another. So in that case, you can see that the bacteria also are able to spread the the antibiotic resistance genes by the help of transposons. So transposition happens only inside the cell, but the result can uh, be that they transfer specific genes from chromosomes or from one plasmid to another. And if this plasmid is transferred into another bacterium, it means that they have spread those genes. Okay, so I'm done with this lecture. Do you have any questions? Do you have any questions? Okay, so if you don't have any questions, I'll see you at 1. Let's take a break and see you at 1. Thank you for your attention.